Hi, my name is Lilit Antonyan, and I am a PhD student at McGill University. I am currently working at Dr. Carl Ernst's lab at the Douglas Institute in Montreal, Canada. So the way I got involved with the SETBP1 disorder research is that I joined my PhD degree in 2018. And around that time, my research group was in communication with Haley Euler, who's the president of the SETBP1 Society. And they were discussing about potentially starting a new project to research SETBP1 disorder. So I just jumped right in. So I'm working on understanding how the loss of one copy of SETBP1 from our genes can lead to disease by using brain-like cells, and this is something that we call disease modeling. So what we do is that we collect cells from children who have SETBP1 disorder, as well as their healthy relatives, to use them as a comparison, and then we grow their cells in a dish. And then once we've grown their cells in a dish, we transform them into stem cells. And stem cells, they have the capacity to become any cell that's found in our body, like cells from our eyes, or cells from our heart, or cells from our kidneys. So we take advantage of these stem cells and we convert them into neurons and other types of neural cells. And once we have these neurons, then we can look at the way they communicate with each other, the way they're growing and the way they're maturing, the way they communicate signals inside of the cell and other functions like this. I think it's really important to research SETBP1 disorder because it is a rare disease and there is very little information known about it. And I can only imagine how frustrating it can be for a family who has a child that is born with SETBP1 disorder, and there's not that much information to go on. And so families don't know what to expect in the development of their children or how they can provide them with the appropriate tools to reach their full potential. So I think once researchers such as myself and doctors can better understand how the disease works, then we can focus our efforts in order to find a different therapeutic avenues to help these children who have SETBP1 disorder. So currently, with my research, we're interested in understanding how signaling or how instructions are communicated inside of the cells happens in neural cells from children who have SETBP1 disorder. So what we're currently looking at is at the way SETBP1 interacts with its binding partner, which is called a SET, and how they together put the brakes on this important master regulator inside of the cells that's called PP2A. And so PP2A is very important in our body because it can activate or inactivate a bunch of different proteins, not only in our brain cells, but also in different cell types throughout our body. And so the idea that we have is that when one copy of SETBP1 is lost, then this break that typically happens on PP2A is not as efficient as it should be. So then there's too much activity of PP2A. And then we think this is the mechanism that is leading to the symptoms that we see. And so our project goal would be to find different molecules that can restore the level of PP2A to normal levels, and then this can be a therapeutic option for children who have SETBP1 disorder. So I'm currently excited about a lot of things. One of the main things I'm excited about is that how much technology has moved forward every year and how this increases our capacity as researchers to do our research with great tools in order to find answers faster. I'm also excited about how the SETBP1 Society has gone so far in its 3 years of existence. So now we're a really big community, and that's really exciting.